Hello, my name is Roxana Sida, and today I'm going to talk about my 2017 summer research project, exploring the different interaction pathways of ASB9. ASB9 stands for ankyrin repeat in SOX protein 9. Here is ASB9. This region, it's called the SOX box, and this region is the ankyrin repeat domain. Together, they play a major role in targeting proteins to degradation. This means that they bind to other proteins to kill them, specifically to creatine kinase, which is another protein that can be found in cells in the heart, brain, and skeletal muscles. Elevated level of creatine kinase, or CK, can be found after having a heart attack when the heart muscle is damaged or in conditions that produce damage to the skeletal muscles. So it is very important that the amount of these proteins in our body is regulated. That's where ASB9 takes action. This image shows how ASB9 binds to creatine kinase and targets it for degradation by the ubiquitination process. While we have a model for how ASB9 binds with creatine kinase, we are not sure how the dynamics of ASB9 impact binding to creatine kinase, and that's the main objective of my research, to understand the protein-protein interactions of ASB9 and creatine kinase. My hypothesis is that the more extended the residues from ASB9, the more solvent accessible the residues that bind with creatine kinase, and thus the better ASB9 bonds with creatine kinase. We propose here the combinations of these methods to test my hypothesis. First, by using PubMed clustering, we should be able to make an ensemble of the binding site shapes, and then perform clustering and PCA analysis. The clustering part will help us to find metastates binding shapes, and the PCA will help us find correlated subpockets in binding sites. Since proteins interact with each other through their surfaces, if we perform a PBS electrostatics analysis, we should be able to see the surface charges like in the picture. The blue areas are the positively charged and the red ones are the negatively charged. That will give us a valuable insight on where the interaction of CK could happen according to the charges. Lastly, with meta-PPISP, we should be able to predict the interaction site. Generally speaking, you plug your PDB to the meta-PPISP webpage and it will return to you via email the probability of interaction of each residue, which is extremely helpful. I would like to give special thanks to Dr. Romeo Morrow for giving me the amazing opportunity to participate another year in BioChemCoRe and for being very supportive and kind to me. To Dr. Jamie Schiffer for being the best mentor in the world and for her patience. To Jeff Wagner for helping me to set up my analysis methods, and everyone in the Amaro Lab for being great mentors and friends too.